All right, so this is the last part of chapter 3, the energy storage and use. So we're going to start with glycogen metabolism. So again, remember metabolism means building up or breaking down. So we're going to talk about both of those. Glycogen, remember, is the branched polymer of glucose, and glucose is stored primarily in the liver and also the skeletal muscles. The liver and kidney glycogen stores provide glucose for other cells via the bloodstream. So kidney to a smaller extent, liver a big player in glycogen metabolism. So what's interesting is that glucose, but not glucose 6-phosphate, can leave the cell, and only the liver and the kidney have the enzyme glucose 6-phosphatase, which converts glucose 6-phosphate to glucose. Does this make sense? Let's take a look at this. So this is glycolysis. So if we are breaking down glycogen, glycogen breaks down to glucose 6-phosphate, all right? So glycogen is broken down to glucose 6-phosphate. However, remember I mentioned that this hexokinase is irreversible. It does not allow glucose 6-phosphate to become glucose. So another thing is that glucose 6-phosphate doesn't exit the cell. So you can't get glucose 6-phosphate into the bloodstream. You have to get glucose, and then that can go into the bloodstream. So remember also that when you're breaking down glycogen, you're starving. You are trying to liberate the glucose molecules from glycogen, get it into your bloodstream so that your other cells can take up glucose. All right, so glycogen does not break down to glucose, it breaks down to glucose 6-phosphate. But it cannot be made to glucose in most cells because hexokinase is irreversible. And glucose 6-phosphate is not transported out of the cells. So 2 organs, liver with big glycogen stores, and your kidney, have the enzyme glucose 6-phosphatase, in red here, that can convert your glucose 6-phosphate into glucose. So only in the liver and only in the kidney can you do this, right? Glucose 6-phosphate to glucose. Then in the liver and in the kidneys, glucose is created. Glucose can be transported out of the cell and into your bloodstream, thereby elevating blood glucose levels for the entire body, okay? So your skeletal muscle also has really big glycogen stores, but when it breaks down glycogen, it just breaks it down for itself. So the glycogen that's broken down, the skeletal muscle can use it for energy, but it doesn't get liberated out into the bloodstream, okay? So basically the liver and the kidneys are the only organs that can actually donate glucose to the bloodstream. All right. I think that's it. So this is just the overall. So we talked about breaking down glucose, which is called gluco. I'm trying to pronounce this. Glycogenolysis. So glycogen is here. Sorry about that. I got disrupted. Sidetracked for a second. So glycogen lysis, right? So we're breaking down glycogen to glucose 6-phosphate. There's pathways where that can be made into pyruvate. But we talked about how glucose 6-phosphate here can be converted to glucose with this enzyme in the liver. And then through normal cell respiration, right, hexokinase is the enzyme that takes glucose and makes it glucose 6-phosphate to eventually become pyruvate, right? Because this can eventually become pyruvate in glycolysis. What about building up glycogen, right? If we want to create glycogen, glucose is broken down to glucose 6-phosphate. Glucose 6-phosphate can ultimately build your glycogen. So glycogen is actually this molecule hooked up together, okay? So that's glycogenesis, All right, glycogen, glycogenesis, and then glycogenolysis. All right, next topic is gluconeogenesis. So what about making glucose from scratch? What about just making a complete monomer or monosaccharide glucose from other molecules? It's possible. The substrates that are used are glycerol. So remember glycerol? Glycerol was that 3-carbon component of our fats, our triglycerides. So glycerol was that head portion. We can also use lactate or lactic acid, okay, which we see as what's made with anaerobic respiration. We can also use some amino acids. And of course, pyruvate, we can reverse that process. It occurs mainly in the liver, okay, to make new glucose. So again, remember that new enzyme, glucose 6-phosphatase, needs to be used here. That's only in the liver. So the liver is able to make new glucose. All right. Metabolic pathways involved in gluconeogenesis. So you can see, this is just a flow chart of the things we talked about. So proteins can come in. Proteins can actually be made into glucose. So we're looking to come to glucose here. And some amino acids can, there's a keto acid intermediate, and then we have some of our amino acids becoming pyruvate, and then pyruvate entering into the reverse glycolysis. And then we also have some amino acids taking another pathway through the Krebs cycle. Don't worry about these terms, but overall amino acids can be made into glucose. Okay, so gluconeogenesis. All right, what's the answer? It's C, right? Makes glucose from glycerol, lactate, and amino acids. All right, how about energy storage? So our bodies store most of our energy as fat, otherwise known as triglycerides, right? So remember a triglyceride, a glycerol head, 3 carbons, 3 fatty acid tails, which is this one. Lipids have greater energy per weight than carbohydrates or proteins. So there's just a lot of electrons, a lot of potential energy in fat, greater than carbs or proteins. And then most of our fats, our triglycerides, are stored in adipocytes, our fat cells. And this is a picture of some adipocytes. When you break down fats, it's called lipolysis, right? Because fat is a lipid, lipolysis. And so your triglycerides plus your lipases are the enzymes, right? Because ASE, there's our ASE enzyme suffix, breaks it down to glycerol and the 3 fatty acids. So glycerol detaches, our 3 fatty acids detach. Glycerol can be used, this is a very long word, don't worry about that word, but it can be used to undergo glycolysis, and it produces 20 ATP. So that's a large amount of ATP for fat. If you think about the number of ATP made in the electron transport chain was 28 per glucose molecule. It's 20 just with the glycerol alone. And then the fatty acids of the fats are broken down separately in a process called beta oxidation, 2 carbons at a time. And the fatty acids can be converted to acetyl coenzyme A. That enters into the Krebs cycle. You are creating NADH and FADH for the electron transport chain, and you get 17 ATPs from every 2 carbons that are taken off. So remember, those long fatty acids can be many, many carbons long, 12 to 15, 16, or it's actually usually an even number, but 12, 16, 18 carbons long. So that's a tremendous amount of energy that comes from those fatty acids. Beta oxidation, by the way, happens in the mitochondrial matrix, the same place where the Krebs cycle happens. Okay, so taking a look at this, the pathway of fat breakdown or lipolysis. So here is our triglycerides, here's our fat. we are breaking it down to both fatty acids and glycerol. Glycerol can enter into glycolysis, all right, and it can form pyruvate. The fatty acids are broken down through beta oxidation. They enter into the Krebs cycle, and you can see that it basically feeds into this whole central process of ATP synthesis. So your fats all come into, eventually the ATP is made through oxidative phosphorylation. So they all contribute components to that central pathway we talked about. I highlighted ketones here because there is this fad diet out there. Well, I don't know if fad is correct, but it's been a long time coming. There have been protein-rich, low-carb diets touted for decades, starting from the Atkins diet, and now it's called the keto diet. But what happens is you restrict your body of carbohydrates, and so your body needs to turn to fats and eventually proteins to, you're starting to break down fats as your energy source. So when you break down a tremendous amount of fats, what builds up as a byproduct are these ketones. We talked about this before in another lecture. So these ketone bodies are acids, and so they can cause your blood pH to drop, and that is called ketoacidosis. Right? So be careful with low carb diets because it can be dangerous. Moving on to your protein metabolism. So when you eat proteins, remember that your proteins are made of amino acids, and that central carbon had an amino group which contained nitrogen, and a carbolic acid, a hydrogen, and an R group. Okay, anyways, when you process your proteins, you are going to break them down into amino acids, right? So that's proteolysis, protein lysis. When you break down the amino acids, you're going to take away the amino group. So that's called deamination. The amino group is removed, and guess what's created? Keto acids and ammonia. Again, if your body is forced to break down fats and proteins as energy sources, keto acids can build up to dangerous amounts. Ammonia is produced. That's not good. So ammonia actually travels into the bloodstream. Okay. Ammonia can be converted to urea, it's converted to another molecule called urea, less toxic than ammonia. And then the urea is excreted in your urine by the kidneys, right? And so the more protein that you eat, the more urea is in your urine, because your body only uses so much nitrogen-containing compounds. If you're not actively building muscle or using a lot of protein, then if you eat like a really big protein-rich meal, your body's not going to use all of those amino acids. And so it'll break those amino acids down into urea, and urea is going to be excreted in your urine. All right. So just looking at this picture here. So here's our metabolic pathway for proteins. So proteins broken down to amino acids. Amino acids can be broken down furthermore. The amino group can be converted to urea, and this is excreted. Keto acids are made as the intermediary, but under normal circumstances, if you have all these different food groups and not a tremendous amount of protein breakdown or not a tremendous amount of fat breakdown, your keto acids are converted into harmless molecules. So they can be converted to pyruvate, it can be converted to acetyl-CoA, or some kind of intermediate of the Krebs cycle. Okay, and so we can, keto acids are a natural part of our metabolism. We just don't want it to build up to toxic levels. All right, so I think we may have reached the end of our lecture. And so that's it. That was the end of part 3.